Okay, so today I will continue with the calcium dynamics that we started last time. So I gave you these three definitions. So I told you if you want to basically, after you get the signal to your muscle, you want to do something, right? So the muscle will contract. And the way that you have this cell signaling and so on in the muscle is through calcium release, right? And within the cells in the muscles, basically you have these two things. So this is within the cell still. You have something called the sarcoplasm, where you have calcium ions. But you also have something more specific, which is where you store the calcium ions, and it's called sarcoplasmic reticulum. And today we will basically write down equations that will model this calcium-induced release. So before I do that, let me give you a few more information pieces that we need. So we have that extracellular calcium concentrations are higher than the intracellular. Calcium concentrations. So calcium must be pumped out. So muscle cells, just give you still some background, are bundles. These are called fascicles, and they consist of fibers. Each of which contains arrays of filament structures. These also have a name. These are called microfibrils. And these are the ones that contract under the action of calcium. Okay, so basically what you have is a structure like this. So say this is some muscle, has these filament structures. And then you have this calcium now action. And basically, if this was my original configuration, I'm going to have something that contracts. So it's like a bicep or something, I don't know. So let's write here muscle contraction. And think of okay, and basically similar to what we have seen previously in the course, if you have a stimulation of a nerve, cell, you can have these action potentials again. An action potential gets triggered, and it propagates. This time is not along the axon, it's along a fiber in these muscle cells. Okay, and if you look at this experimentally, basically you can look at the release of calcium. So if I draw, this is the calcium concentration, and this is time. Let's say this is concentration. I'm going to get something very spiky, so it's gonna look like this, and so on. So the release, this is saying that the release of calcium is spiky. Okay, and the question is, can we derive now a mathematical model to describe this? So can we derive a mathematical model to describe muscle contraction with a low calcium concentration in steady state that is excitable under stimulus. So there is a model is known. It's called the two-pool model. And basically what you have to take into account now is that you have this intracellular fluid, which is the sarcoplasm, and there is this inner store that we call the sarcoplasmic reticulum. And then we will keep track of the influx, so the uptake of calcium within this little store, the sarcoplasmic reticulum, and then what goes outside of the cell in the outside world, let's say. Okay, so this is the idea. And then once we know all the components that are going in and out of the cell, we will be writing down, using the law of mass action, what the differential equation should be. And once we have the equations, we will do the usual phase plane analysis with null clines and so on. Okay? All right, so the picture that I will draw now is the most important part because it tells you what we are trying to model. So the board is basically my cell, and then I have the muscle cell, and then there is something inside it, which is this sarcoplasmic reticulum. And what is outside here is the sarcoplasm. So it's still the fluid inside the cell. And I will be denoting by CS the concentration of the calcium inside the sarcoplasmic reticulum. And I will also say the concentration of the calcium is C, so without the subscript. So C here is the concentration of calcium in the sarcoplasm, and CS is the same but in the sarcoplasmic reticulum instead. Okay, so other things that you will have here is, so let's draw it with green maybe. There is some leakage again from the sarcoplasmic reticulum to the sarcoplasm. This is some active release. But I will also be having an intake, so I'm going to denote that from the sarcoplasm inside here is going to be J+, and this is an active uptake. Okay, so let's write this down. So J+ is the rate of take-up of calcium by, so this is SR, by the way. I'm just going to use this so that I don't have to write it all out. And this usually happens by something called a receptor. Okay, so this is my active uptake. And J minus is basically the rate of release from the SR to the sarcoplasm. And this is the active release. But we will also have some kind of diffusion. So that will be proportional to your concentration of calcium inside here and in the sarcoplasm. So there are two extra things. This goes outside. As I said, it's proportional to CS. So I'm going to write KS CS here. And then I'm going to get some from the sarcoplasm to the outside world. So whatever happens outside of the muscle cell. So let me denote it here. I can have something which is proportional to KC, and it's also a leakage term. And finally, apart from this, there is also an influx. So let's see where I will draw it. It's like this. That comes from the outside in the sarcoplasm. So this is an R influx. Okay, so I will write them now over here. So R, the last thing I wrote, is an influx of the calcium concentration into the sarcoplasm from the outside world. And then it's because basically you have applied some external stimulus of an external. And the last two things I need to define are the leakage terms. So I have also KS CS. This is the rate of leakage of calcium from the SR to the sarcoplasm. And this is something that is, so notice that in the green ones I wrote active release or uptake. In this case it's just diffusion, so that's why it's proportional to the concentration. It's something that is passive. So this is passive and it's proportional to concentration. And the last one is the KC, which is the leakage from the sarcoplasm. So it's the same as above, but it's from the sarcoplasm to the outside of the cell. And it's again passive. All right, so this is all you need. Now I will use the law of mass action sort of to basically write down the two differential equations we have. So the differential equations that we will need are how the concentration CS is changing and how the concentration C in the sarcoplasm is changing with time. Okay, so we'll have two differential equations. And those are given by the following. So I have dCS dt I will define these two things. They are formulas. And I will also use here F because I will be using it later, so I don't want to keep cutting this. So this is capital F for us. So this is saying how is the concentration CS changing? So I have J plus is adding to it. I'm removing an active release of calcium, right, from inside. So this is minus that. And then I also have diffusion that is proportional to my concentration inside. So that's why you have J plus minus J minus minus that. And then can anyone see what the dc dt will be? So first of all, how does it relate to whatever I have written for dcs dt? Those are terms that are being gained or lost from the sarcoplasm. Yes, exactly, it's lost. Perfect, exactly. So I will have minus df basically. And what am I adding to the sarcoplasm? Additional things. Yes, perfect, so I should write R, right? Yes, and you subtract this, exactly. So it's going to be R minus the leakage thing, and then minus that. So is it okay if I just write F? Okay, so now we have to choose together what these two should be. And basically, it's again experimentally, but they have the form of a Hill equation. We did it in the first lecture, maybe. So the form of the active uptake and release is this. So let me first write it down, and then I will define these things. This, again, you find from experiments what the value should be. But here I have that these numbers are just constants. So the V1 and the N, these things are all constant. Only the C is the concentration, so these are all constants. And this thing, if you remember when I did in the first class the Hill equation, this is of this form. So this is a Hill equation. So this V1 is not denoting a voltage like we were doing in the previous lectures. It is a concentration rate. And I have that all these things, let me just write it here, V1, K1, and N are all just numbers. And I also have to define now a similar thing for J minus. So J minus is defined, it's a bit more complicated in the sense that it has a term that looks like that, but with V2 and K2. So this is K2 now, N plus, and notice that it's CS, not C. And this should be M. And then I multiply this by something that depends on the concentration, C. So it's going to be C to the P divided by another constant K3 to the P plus C to the P. And we will have that this is again another Hill equation. And these two parts are not as important. So these are not that important. And the thing that matters is basically this, and I will tell you why. So this second term is the one that controls, this term controls the calcium-induced calcium release. This is the term controlling the calcium-induced calcium release. Okay, so as usual, in order to be able to say something about the system, I need to do a non-dimensionalization, right? So, and then once I non-dimensionalize the system, I will have some important parameters. We will see that there is a fast and a slow scale again, and then I will do the null Klein analysis. So here I will do a non-dimensionalization. I will write everything in terms of U and V again. But the U and the V will be related to the concentrations C and CS. So C, the concentration in the sarcoplasm, will be written as K1, that K1, times U. I will have also the concentration in the sarcoplasmic reticulum CS is going to be now K2V. The time is going to be non-dimensionalized by 1 over K. K is the rate of leakage in the sarcoplasm. And then I will have T hat here, the dimensionless variable. And finally, I also have to non-dimensionalize the F, which is those active releases and uptakes. So for this one, I'm going to use V2, the V2 from the J minus F. So here note that the U, the V, the T hat, and the F are the dimensionless variables. So once you use this in the differential equations that I am hiding, those two, you will be able to derive now two equations for U and V, which are the following. So I will have du dt is equal to mu. The mu will be some combination of the constants that we have there that I will define, minus u gamma over epsilon, and then I have f of u comma v. And the dv dt is going to be just 1 over epsilon f of u v, where this f function is some combination of powers that involve the n and the m in the Hill equation. So it's going to be some parameter beta I'm skipping a lot of terms, a lot of steps here, but you can show this from the non-dimensionalization. And then we have up, and then there is an additional term where you subtract minus delta v, where this and the epsilon are small parameters with the dimensionless parameters. The mu will be really important. Why will it be important? Because when I want to find the null clines of these two equations, I'm going to set these two derivatives to 0. This will be 0, right? For the v null cline. So that will become 0. So it will depend how the u, the magnitude of u compares to mu. That's why I'm saying mu will be important. So, okay, what is the definition? Mu is given by the ratio between r. So I know you're just getting introduced to this topic, but just bear in mind, because I will come back to it. The numerator here is the influx from the outside to the sarcoplasm. This is basically related to the diffusion from the sarcoplasm to the outside, right? And the K1 is inside the J plus. Okay, so just try to remember what it means physiologically, because we will use it later. Okay, so I also have to define gamma. Gamma here is K2 divided by K1. Again, it's a ratio that takes into account something that appears in the J minus and the J plus. Epsilon is also important. So this is K2 divided by V2. It's important because this is a small parameter. And then I also have, this is K3 divided by K1. Beta is the ratio of the V's. And then finally, the delta is also a small parameter given by Ks K2. And this is much less than 1. So I'm going to highlight these three. And also I want you to remember that U is the thing that is relating to CS, right? We said. And then the V is the one relating to, sorry, it's the other way. V is related to CS and U is related to C. Okay, so how does the alkaline look like? So this is the first thing I want to do. This is now a 2D system. That involves the variables U and V. So we will use, as usual, phase plane analysis. So this tells us that since the epsilon parameter is very small, I will have that we will quickly move to the V alkaline, which is given by f of u comma v is equal to 0. So that looks very complicated, right? The function of f. But we also know that delta is also a very small quantity. So you're going to set that to 0. And you're going to have a combination of these powers of u and v. But we will want to know how to plot this, right? So basically, I will first plot it, and then I will walk you through the steps of figuring out how to do that. So this is v versus u. And it will look something like this. This is f of u comma v is equal to 0, which is the v alkaline. Okay, so the question is, how do we plot and I believe that this will also appear in your problem sheet. So basically, I will walk you through it up to a point, and then I will stop, okay? So how do we plot this? So first, I said that the delta is much less than 1. So if you ignore the delta term in the f equation, the delta term in f of u comma v equals to 0, you can get the following. You basically have f is equal to, and then you have those ratios of u and v. So let me write it in exactly the same way. This was 1 plus u to the n minus, and we had v to the m divided by 1 plus v to the m. And finally, u to the p divided by alpha to the p plus u to the p. There is this, I'm setting it to 0, and this is equal to 0. So now that I have this is equal to 0, I can just say this thing, basically I will rearrange it for v, and I will get something that is this divided by that. So it will just be v is equal to some function of u. Does that make sense? So I get basically v to the m over 1 plus v to the m is equal to u divided by u to the p. This is a power. Alpha to the p plus u to the p. And these things I will define as two new functions. This is going to be my j, and this is the k of u. Did I forget? I forgot the beta, right? Okay, and basically the way you think about plotting this is the following. This you will know what the form is. It's basically like a sigmoid function. So if you draw that, it will be something like this. This is your k of u. And then you have some similar form for j. There will be some intersection point uc. This is u. These are monotonically increasing functions. And then you can also draw the ratio of the two, and that will be of the following form. So here I have j of u divided by k of u versus u again, and it has this form. Now you can solve for v exactly. So if I just multiply this by that, and this by this, and then bring all the v to the m terms on one side, factor out, I'm going to get an expression for v just in terms of u. So I'm not going to write every single step, but basically from rearranging, you get 1 plus v to the m, j of u, divided by k of u, and then this gives me v is j divided by the difference between these two functions raised to the power of 1 over m. And I will give this a new name. This will be phi of v. Okay? So if you now draw something like this is actually phi of u, sorry. It's u functions. So phi of u versus u, it will look like this basically when you draw it. So this will only give us one of the sides of the function. Remember that what we are trying to plot is v versus u, which as I plotted before, it looks something like this. This is the u, the v null line. So to approximate what is happening here, you're going to take into account the delta parameter being small. Set v is equal to v divided by delta. So in order to do that, there is also extra information in the typeset notes for how to do that, okay? All right, so maybe I will say here so that you remember, see problem sheet. Okay, so once we have this now, we want to look at how the dynamics behave. So the next thing is, so this is where we do the phase plane analysis. Look at the dynamics. So, okay, how are the equations? So let me rewrite the u and v. So that was du dt. You don't have to rewrite it. It's just so that we can see it. Is mu minus u, and then I had minus gamma over epsilon, this function. And then the dv dt was easy. It was just 1 over epsilon f of u comma v. And we said that basically now, what we can say is that because of the epsilon in the dv dt equation, we have that v rapidly approaches the null line. But there is also the epsilon in this equation over here. And since I have this, I can now rearrange it basically, or like plug it in rather, and write this as du dt with something involving dv dt also, right? So if I take this thing and I write it as epsilon dv dt is equal to f of u comma v, I can take this and plug it in here. So I'm going to have a combination of u and v together in one equation. So this gives me du dt. If I bring it to the other side, it's going to be plus gamma dv dt. The epsilons cancel, and I'm left with this, correct?
Why does it rapidly approach the null line?
Because this is gonna be small, so it's gonna be f of u comma v is equal to 0. Yeah. It's okay? Okay. So once I have this now, gamma is just a constant, right? It's our dimensionless parameter, which I don't know where I wrote anymore. Sorry to do this. I think it was here. Yeah, gamma is just the ratio between these two things that appear in our fluxes. So basically, you can think about doing the following. This is the same exactly as writing d dt of u plus gamma v is equal to mu minus u, right? And now I will do the following again. So I will do a rescaling. So if we have a fast time scale, we can write t is equal to epsilon tau. And now just replace wherever you see d dt this by having d d tau now. There will be an epsilon term that appears. So from this now you will have basically d dt, d tau, sorry, u plus gamma v is equal to epsilon nu minus u. So what this tells you is that to leading order in epsilon, you're gonna have u plus gamma v is equal to some constant. So this is in leading order in epsilon. So if I rearrange this again, I will have, so let me think, I'm doing v versus u. So I'm going to have minus 1 over gamma u plus const divided by gamma. Okay. So if you're plotting now the the v versus u, you will have these points. These are like tangents, right? Because if I take the derivative dv du, I'm going to have some gradient minus 1 over gamma. Right? So that will be useful in what I do next. And this again is very similar to what we were doing in the Fitch-Nagumo analysis. So I have my alkaline. It looks like this. This is f of u comma v is equal to 0, u and v. And I have now 2 points on this curve. These are mu minus, and this point over here is mu plus. And I will define them over here. So mu plus and mu minus are points where the gradient of the curve f of u comma v is equal to 0 is minus 1 over gamma. So it's basically, if I draw now the gradient of this, let's see how I do this. And this will be basically another line that hits here with the same slope, more or less. Maybe that curve should not be exactly drawn like that. Okay, so these are, if I take the slope of this, it will be minus 1 over gamma. So this is v is equal to minus 1 over gamma plus some constant. And then this also. Okay, so now I will look at 3 different cases when I'm doing this phase plane analysis. So the first case is when I have a mu that lies in between these 2 intersection points. So I have mu is between mu minus and mu plus. And in this case, if I go back to my equation, which is this one, but with the t, I have the following. So just imagine that there is a line that lies between the 2. This is where mu is. And we are trying to analyze the derivative. So I have d dt of u plus gamma v is equal to mu minus u. So we have basically now two cases. I have to look at when u is greater than mu. So I'm to the right of this line, but before mu plus. And I also have to look what happens to the derivative when u is less than mu. So you just have to look at the signs of this derivative and see if it's increasing or decreasing. So if u is bigger than mu, this will be negative, right? So I'm moving to the left. Does that make sense? If this is less than mu, then this is positive now. It means this is positive, therefore this is increasing. So I'm going to be going to the right. Yes? Okay, so we move to the left, and then if we are less than mu, we move to the right. Okay, so if I want to draw that on the picture there, I'm basically on the alkyne, and I want to see if I'm to this side, I'm going to go left. Let's see. Okay, basically I also have this, but I will draw them later. So u, which is this axis, is bigger than mu, but mu is between that, so I'm less than that. I'm going to the left, so this is this arrow here. And now u is less than mu, so I'm to the left of the red, but I'm to the right of mu plus, and I'm going to the right. So this is the arrow here. Okay, so I told you there are two more cases, so I don't have space there. Let me draw it here. The next case that I will do is now mu, this red thing, is going to be to the left of my mu minus. So I'm going to be looking at this region over here now. So case 2, the mu is less than mu minus. And I will do exactly the same thing. So basically I take my differential equation over there, which is u plus gamma v is equal to mu minus u. And I will have to look at what happens when u is less than mu again, u bigger than mu. Okay, so u less than mu, it means that we are moving to, again, you do the same thing. U is less than mu, I'm going to have something positive. We move to the right. And then when we have this, it's left. So if I come to the picture now, maybe I will draw it again so that it's not too crowded there. You have the following instead. So I have V, U, I have the V in alkyne as usual. This is, it doesn't look exactly the same, but it should be. Just pretend it's the same. So I have this. And then I drew those things with the green before. So I have these two curves. And now what I'm saying is that if this thing is mu minus, and I have also the mu plus, my blue or red line over there is going to be to the left. So I have it here. So this is mu now. So what we just found is if u, this axis is to the left of mu, so I'm in this very small region over here, I'm going to be moving to the right. So I have an arrow that goes like this. Maybe I will draw it with a different color. I'm sorry about this. This is like that. And then I'm to the right of mu, but still mu is less than mu minus. So I have to be restricted in this very small region over here. So you go left there. You see what I mean? So there is the fixed point, which is the intersection of these two alkynes, basically, which is here. And then I'm looking at what is happening in this very small region over here. And I'm saying I will basically be trapped in this equilibrium point. You see what I mean? Okay, great. Okay, so the last thing we have to determine in the last 5 minutes of class is what happens in this very small region here. And to determine this region, now you look at what happens when your mu is bigger than mu plus. And either you have to go out or in again. We will see depending on the sign. But what is the physiological meaning before we do that of this? So I have to tell you what happens in this case. So when mu is less than mu minus, this says that we need a bit of energy or some excitation in the cells in order to move away from this equilibrium point, which is the blue. And this is in order to get an excursion, which would be the muscle contracting. Instead, what this region that I was telling you about before is not the muscle contracting, it's instead something that you will see we will get trapped in that small region, and it will be like a cramp, basically. Okay, so the calcium cannot be released. This is what this will be saying. So this is the last one, case 3. I have mu is greater than mu plus. Again, I look at the differential equation as before, and I have d dt u plus gamma v. This is equal to mu minus u. And then I have when u is less than mu. Again, this is something that is positive, move to the right. And when u is greater than mu, we move to the left. So the final picture is this. V, u, we have this. This is now, I feel like every time I draw it, it gets progressively worse. This is supposed to be deeper so that I can draw this. Okay, this is like that. And we have the 2 points. This is the mu minus, the mu plus. And then I have a line that is basically in this small region over here. So mu is greater than that. And what I'm saying is that this now, if I'm to the right of mu plus, but less than this, I move to the right. And otherwise, I move to the left. Okay, this is our small region there. Okay, so the physical meaning of this is that calcium cannot be released. So the equilibrium point lies at, now u is greater than mu plus, which is where the calcium concentration is high. And this leads to cramps. Basically, it says that where you have the high concentration of calcium, if you are in that region and you have this equilibrium point and you're going towards it, you will really need huge amounts of energy to escape from that point. Whereas if you are here at that equilibrium point, even if you apply a small amount of energy, you will be able to escape. And if we are somewhere in between, which was case 1, so in this region over here, if I start at some point, it's basically going to lead me to the other nullline, and I'm going to go back to this point. So it's like a periodic oscillation that keeps happening. But because there is no equilibrium point in between, it's not going to remain there. Okay, so the 3 parts have also a distinct meaning. Muscle contraction is here. These are sustained oscillations or relaxations. And then here is cramps. Okay, there is also something called rigor. This marker is dead, which is very fitting because there is also something called rigor mortis. Does anyone know what this is? What is it? Exactly, yeah. So it also happens in that region over there. So it's like the muscles basically get stiff. Okay, so starting next week, yes, tell me. Because basically, if you look at this point over here, and you remember from the Physiology equations that we had that cubic, right? And then what I was drawing is that if you start at some point, my cubic was the other way, right? I think, actually. 1 second. Is that if I start at some point, basically, or was it correct? I don't remember. Let me see. It's very analogous to that. Yeah, no, it's like this, but I just have to draw this here. If you start at some point over here, basically we had that you go fast in here, you go up, right? And then you come back here, and then you have to go back to the equilibrium point, right? So it just has to do with finding the equilibrium points. Where are the equilibrium points for this system? Is where our equations are both equal to 0, right? So it's where this is equal to 0 and mu is equal to u. So what are the equilibrium points for us? It's only when u is equal to mu. So it doesn't happen in that case. You don't have an equilibrium point. So you just go around. Whereas in the other two cases, you will be going towards u is equal to mu. And you can stay at the equilibrium point. Does that answer your question? Okay. So yes, next week and for the following lectures, I will not be the one teaching. But for this first part, I wanted to say, really know well how to do phase plane analysis for your exam. Null clines, all these things. Know Fitch-Hugh-Nagumo equations. Know the traveling wave equations that we did. And yeah, you should be fine. Also, make sure you pay attention to like the physiological meaning of things. That's all I can say. Thanks.